Yes.
Good afternoon, everybody. Kia ora. Welcome to Lincoln University. This is Katie and myself. We're both from the B-Link team here on campus, and B-Link Innovation has been hosting the Excellence series for the last while. Now this beautiful building is finished. We're actually going to move. Our Excellence series in here, but it's our first time. So we're a little bit nervous that will all the tech work? Will everything work? Will everybody show up? Can you actually find the building? You did. Phew. So once again, welcome to Lincoln University. Welcome to the Lincoln University campus, and welcome to the Excellence series. What we'll do is I will do a brief health and safety. After that, I will introduce Vice Chancellor, and then he will be your master of ceremony, your MC for the rest of the afternoon. I'm going to listen to Jim. I'm going to see if this works. Yeah, cool. Housekeeping. New building, emergency exits, right behind you. 2 doors. If there's anything going on, that is probably the easiest way to go, unless there is a fire out there, because Forbes Lawn, that is the piece of grass that you actually see out there, is actually the place where we're actually going to get her. If not, we can use these doors, and you actually go out the way you came in. We have not, we don't have any fire drills, so if we need a fire alarm, it is the real deal. And please follow Katie herself, and we have some other Lincoln University staff that has been initiated in the health and safety of this building. So we should be good. Toilets. You go out this door, you turn right. It's your first entrance, and to your left hand side. For people that are really tall, be careful, don't bump your heads. After this, there will be a networking event. We will be serving some little snacks and drinks, including alcohol. Please consume these responsibly, because your home front, your whānau, wants you home safe. So without any further ado, I would like to introduce Professor Grant Edwards, Vice Chancellor of Lincoln University.
Good evening, everyone. I think the first task for me tonight is a reasonably sad one, and it's to make you aware of the passing of Emeritus Professor Andrew Sykes. Andrew Sykes was a professor of animal science here at Lincoln University, and was also a former Vice Chancellor of this university. And he died at his home in Brookside under the care of his daughter and his sister on Tuesday night. I think many of us in the room had an association with Andrew Sykes, whether it was at the time as a colleague in research, or as in time when he was head of department for us over an extended period of time. And so it is with sadness that we make this announcement. Andrew's always had his prime role as a teacher and a research supervisor was to ensure that Lincoln University graduates in animal science could operate at an acceptable international level, scientifically, and to go to any university or research organisation in the world and not be out of their depth intellectually. This is his legacy when he left Lincoln and retired from Lincoln University in 2012. I just wonder if we please take a moment's silence just to reflect on Andrew Sykes, please. Thank you. Thank you. Moving on. Tēnā koutou katoa, and welcome to the Te Whare Wānaka Aoraki Link University Excellence Series. And I'm pleased to welcome you to the sixth in the series, in the Excellence Series. It is through our Excellence Series that Link University is privileged to be able to contribute deeply to public debate in many critical areas facing the country and our communities, while showcasing the university's world-leading education, research, and the land-based sectors. Today is also very significant as it is the first session in our flagship science facility, Waimarie. So the teaching block has been opened here, and progressively over this next month, the research and office and other space and postgraduate space will be opened at the back of the building, with the grand opening of this building on the 27th of September at 10 a.m. in the morning. It is an opportunity to showcase this building, as well as some of the research that will be conducted in this building through the lights of the presentation tonight. In this evening's presentation, we welcome Jim Morton, where Jim will share his work exploring the role of proteins in living organisms and how they are modified in both life and how they are turned into products. Jim is a biochemist and Dean of the Faculty of Agricultural Life Sciences. He has studied proteins in bacteria, plants, and animals for 40 years and has particular interest in the modifications of proteins of economic interest. I personally remember Jim when I was doing my biochemistry second year at Link University as part of my Bachelor of Agricultural Science, and Jim was my tutor in the animal biochemistry class. And I've had a long association with Jim as a colleague and somewhat as a mentor over that time period. Jim's work has included sheep models of cataracts, protein modification to meat quality, and including the environmental and health aspects associated with meat. We welcome Jim Morton to this presentation. Thank you.
Kia ora koutou. Thank you everyone for coming. And I think thank you, Grant, for that introduction. Today, I'm going to be talking about, I'm going to start off by giving a little bit of that history that Grant referred to, and then looking at some of our work, particularly around meat, but some of the other proteins. So, just to start off with, just to spell out why I'm a protein biochemist and why proteins are important. They're really the part of living organisms, the molecules that actually make things work. They're the machinery of life. So proteins are made from codes on DNA through RNA, and they're made on ribosomes. And you end up making a protein, and those proteins make all those other things, and they make everything else. So it's proteins that make the lipids and carbohydrates and everything else that makes this up. So the interest in proteins is because of that. And if you look at animals in particular, most functions in animals are carried out by proteins. And about 20% of animals' weight is protein. And also within plants, proteins are really important, although they have a lot more work done by carbohydrates. But the requirement of plants for nitrogen, which is really important in an agricultural sense, is largely because of the proteins in the plants. They're also an essential component of the human diet. So, without protein, you can't just survive on calories, you need to have proteins. And importantly, in our environment, in New Zealand, there are a very large percentage of our exports. So our goods exports are absolutely dominated by proteins. My experience with proteins goes back, as Grant pointed out, 40 years. I started in Dunedin working on an enzyme in a bacteria with George Emerson, actually, who was Richard Emerson's father. And then when I came to Lincoln, I actually worked on proteins in plants. And it was actually rust fungi, which means that when the faculty merged with fire protection, I actually felt I knew something about what we were talking about. And I also worked for a long time with Mitchell Andrews in particular on nitrate assimilation in plants. So I have worked in bacteria and plants, but for the last 20 years, my work's been dominated by work on animal proteins, and we'll be moving on to those for most of what happens the rest of it. But one of the things that's happened over that time is the technologies we use changed, and what we can talk about in terms of proteins has changed massively in that time. When I did my PhD, this was effectively state of the art. This was separations of proteins. So these little black spots on here are different proteins out of barley. And this is a map of the barley after it's been affected by rust. And these different spots were proteins that we knew had changed after the rust hit it. But we had no idea what they were. We knew they'd changed. Now we have technologies like mass spectrometry in particular, and there's a nice group of AgResearch people down the back that I've worked with over the years that actually use this technology a lot, and we've worked with them, and we can now, each of those proteins, we know what they are, we know their sequence, and we've also moved on from knowing the sequence of amino acids to actually finding out that some of those amino acids have changed. So this is actually, this diagram here shows a separation from a peptide that was part of Anita Grosner's PhD on keratin peptides. And this amino acid here has actually been altered from its original tryptophan to kynurenine. And that's important because those modifications change how the protein operates. It also can change what happens when we eat that protein or do other things with it. So those modifications, many of which occur in living organisms, but some of them will also occur as we process proteins, change the quality of the protein and its various characteristics. Okay, so this next section, from here, I'm going to spend a few minutes talking about this enzyme, Calpine, because this is an enzyme that I've worked for the last 30 years. And it's an enzyme that's found in muscle. And it was first detected because it's found in muscle, of course, turns into meat after an animal's dead. And this enzyme has been found over the years to be responsible for aging of meat. And aging of meat is the process whereby if you hold meat at refrigerated temperature, it becomes more tender. I'm going to talk about that more as we go along. But this particular enzyme, because enzymes aren't put into organisms so we can get tender meat, they actually have to have a purpose in the animal. And this enzyme, what its purpose is it's called a protease. It takes other proteins and chews them up. And the proteases that you're probably most used to are things like trypsin and chymotrypsin, which are in our gut and we use to break down our food. Calpine's quite a specific protease. It only attacks very specific proteins, and it's controlled by calcium. That's where the cal comes from. And so it only operates when calcium levels are high. And it's used when we want to remodel ourselves. So typically it's used when we're trying to change things. And in muscle, it's largely used after development of the muscle in muscle repair. So it's used when we're trying to fix the muscle up again. After the animal, so interested in that in general, what happens with it in meat is it has a specific role, as I said, in tenderness of meat. And in the 1990s, I started working on this enzyme because I realized, being at Lincoln, that red meat was actually one of the more important things in the New Zealand industry. And I was working with Professor Roy Bickerstaff, who's over here handing out meat back in the general. And red meat's our second most important goods export after dairy, and it was worth $9 billion in 2022. And one of the things of meat quality is that tenderness is the most important determinant of repeat buying. So people select meat on its color, fat content, things like that. But you can't tell how good the meat is until you eat it. And tenderness is one of those characteristics. They found that that was a thing that made people come back. So if they're a good steak, next time they come back for another one. So we're really interested in this tenderness. And as I said earlier, becomes more tender with time. And calpain is the enzyme that causes that, because after the animal dies, the calcium in the muscle actually is no longer controlled, so it increases when the calpains become active. And that process happens naturally, but you can actually alter it. So things you do before the animal dies, things you do after the animal dies can change that process. And we've done lots of different experiments to actually optimize that, because what we want to happen is that process to happen consistently, so you get good meat, but also relatively quickly, because you don't want to be hanging around for a long time with it at chilled conditions. So we did a lot of those studies. I'm going to just work on one of them, though, to show you one of the more recent ones. And that was work with this machine here, which is a high pressure processing machine. So normally in New Zealand, we just hang the meat for a reasonable amount of time. And if we're using chilled meat, we're sending it overseas anyway, so it will age on the boat. But sometimes we're interested, and if we've got prime animals, that will lead to quite a tender product. And New Zealand meat is widely regarded because it does that. But particularly if you look at our beef herd, we have lots of animals that get in the beef kill who are not designed to be meat animals. And that's a lot of the dairy cow animals and other older animals. And so we were interested in methodologies that would make that meat tender. We were also interested in whether we could shorten the process of tenderising meat. And this high pressure processing does that. So what we were looking at, we were taking different lots of meat, we're putting them through this machine, and then we were measuring the outcome. And so we were actually testing the effect of 28 days aging, so keeping it refrigerated for 28 days, 1 day aging, which is when it normally be tough, and also a couple of pressures in the machine, so 170 by 250. And what we found is we got dramatic improvements in tenderness. So you can see the 28 days here, and you can see compared to the prime, it actually, for all of it actually brings the tenderness, it makes it at least as tender as 28 days aging. And in fact, for the prime, it made it even more tender. So we can get the same effect by aging, but we can take 27 days out of it by using the high pressure processing. And we also tested that we didn't change the color, because sometimes high pressure does. And we also tested whether the consumers found that it tasted good, because you can make meat tender by mushing it up. So we were interested to make sure they still had the taste. And it came through well on the taste testing. When we actually looked at the product, we found we were actually causing mechanical changes, because this is normal meat after aging at 1 day. And you can see the structures. Those of you who are used to muscle will be easy to see, but everyone should be able to see there's a irregular pattern there with the individual sarcomeres. And these are these lines here between these lines. So this is a pattern that's repeated right along the muscle. And when we compared it to our meat that had been through the high pressure processing, you see that here, this has been contracted quite. So there are areas of extreme contraction, and we've lost this pattern. So what was actually happening was the high pressure was activating the meat to actually contract quite strongly. It also was activating calpains. And the combination was leading to this tenderization. So a lot of our work has been along that basis, trying to make New Zealand's meat of higher quality and more consistent, because all our meat has to travel along, well, 90% of our meat, the stuff we export, travels a reasonably long distance, and so we need to be getting premium prices for it. Okay, now for a total change, calpains are also important in other tissues. And this is another project that I spent about 8 years on, was looking at calpains in the lens. And so the lens of our eye is another very protein-rich tissue. So we always associate muscle with high levels of protein. The lens has much higher levels of protein than your muscle. And your lens, of course, sits at the front of your eye. And like the lens in a camera, what it does is it directs the light to the back and focuses the image. The key feature for a lens is it has to be transparent. And so to do that, these proteins are particularly well stacked up, and so are the cells. If you look at the cells, it looks a bit like an onion if you cut across them on a large scale. So these cells will go all the way into the middle, which is just the point of the diagram. They don't show that. But. And you can see this is the pattern when we cut across it. So this is one of our lenses when it was chopped down a microscope. You can see this very regular pattern. If that gets interfered with, what you get is, sorry, back to this one, is a cataract. And you can see this is an example of one of our sheep. So this is a sheep with a cataract. You see the lens, this is a bad case. You can't see through it at all. And so cataracts obviously are a major health issue, and it's the most common elective surgery in New Zealand is cataract replacement. And so there was an interest in alternatives to surgery for cataracts. So we were interested in finding a model to study cataracts that an animal model, and that a farmer had actually found sheep that get cataracts in their herd. And because in New Zealand you'd either gone to Massey or Lincoln if you're a farmer, typically this person had gone to Massey. So Massey ended up with these at their vet school, and then we got them down here. And these are sheep that have a genetic disposition to cataracts. It's not a feature of the whole flock. It was originally a Romney, but one particular Romney ran developed this gene. And so these animals, we learnt had high calcium before they got their cataracts, and there was evidence their protein was breaking down. So we thought, well, this looks like what we see in calcium, calpains and meat. And sure enough, it did have. And so we've had them down here, and we studied them, and we found there was this regular progression as they developed cataracts. So we studied these animals, and we found they did have calpain in them, and it was activated. And this is just the model of what we saw as happening. But basically, a variety of insults could occur, and this particular one was genetic, causes a high level of calcium in the lens, activates the calpains, then makes the proteins break down, so the lens becomes cloudy. So what we tried to do, we had 5 years of government funding for this, was working with the University of Canterbury, the really good synthetic chemists, to develop state-of-the-art Calpine inhibitors, in particular this one here, which is a very effective inhibitor, and test them out on those sheep and see if we could slow the cataracts down. Because if we do that, then potentially what you're doing is you're giving people more years life if you can translate it to people. So, because cataracts are typically an old age thing. So if you can actually push them back, then you can avoid surgery. And we did this over a couple of years. We did sheep trials with it. We got that drug working. We got it patented. And we could show a significantly slow progression. And this involved putting eye drops in sheep. And this is Dr. Hannah Lee, who's still working with us now, during her PhD, and there's a postdoc. Actually, you're putting eye drops in sheep day after day, month after month at that stage. And we had success, and we published this. After that, what happens in these pharmaceutical things is then you pass it on, because we don't do human trials. So it went on to University of East Anglia, because they had access to the lenses in UK, the lens bank there. But unfortunately, when we tried to translate it to humans, we weren't able to show significance. So it was one of those examples where we've actually sold cataracts to sheep, but it's not a lot of good. So sometimes this works, sometimes. Okay, so that took me, after that detour, we were back doing meat. We were doing meat through the middle. And just of this part here, I just want to bring up, to talk a wee bit about where we are with meat at the moment, and then some of the particular experiments we're doing. So I'd just like to talk, and I see this as more something that helps the discussion when we get to discussion. But the three areas where meat, which as I've already said, is a very important part of the New Zealand economy, it is also one of the major food groups in the world. A lot of the world has meat as a very significant part of its diet. But if you read our newspapers or listener or watch television, you'll know that meat, there's a lot of concerns around meat and other animal foods, most recently around the environmental footprint. There's also questions around animal welfare, and there are health concerns. So I just want to address some of those and just provide some background to the work that we're doing. So looking at environmental footprint, animals in general are at a higher trophic level. They eat plants, so therefore they will have a larger footprint by definition if you're looking at animal versus plant. When you're looking at food, it gets a little bit more complicated. So typically animal foods will be at the highest level. Some things like nuts can actually get right up there with them because a very small part of the plant is actually the nut. And particularly on a basis of per calorie. So I just want to talk about some of the things to take into account when you look at those diagrams that show meat having or animal products per kilogram or per calorie having a very large footprint. First is when you look at that, you really partly have to look at it as part of an overall diet. So if you look at the impact, you're not really, when people eat, they don't just eat one product. We're omnivores typically. And so we eat meat and other products as part of the diet. So you've also got to consider the percentage in the diet. And also, the calories that are actually in the product don't equal what you actually get inside, because what you get inside is what you are able to digest and take up. And animal products, in particular meat, are often more digestible and more available than plant products. So you get more of what you actually put in. And in particular, what meat is particularly good, it's good at delivering protein, iron, and micronutrients. And that's really important because if you look at the Sustainable Development Goals that just come out again, last year we actually went backwards on the 0 hunger goal. I mean, the last few years have been pretty tough in food production. And 0 hunger is about not only how many calories to give people, we've got a far larger problem with protein malnutrition. So many more people who get adequate calories are really suffering from lack of protein. And this figure, I think, is particularly important. The 30% of women 15 to 49 worldwide are anemic. So iron is actually a really, other than the nutrients after your macronutrients, iron is a really important nutrient. And it's very hard to go past things like meat as a delivery source for iron. So for individuals, these choices, can be made. But at a population level, I think we've got to be very careful to try and take very significant groups of food out of our diet. The other thing I'd say, although, is that when we look particularly at pastoral agriculture, which is New Zealand's normal form of agriculture, it's often on land that you can't then easily turn into crops or horticulture. So we've got to also think in terms of what else will we actually do with that land and where will we replace that food source. And this is also important when we start looking in places like Africa and some parts of South America, where there are those pastoral lands that aren't really that useful for other food production. So from my mind, we should really be looking at making this, if we're going to do this agriculture, we should be looking to make it as sustainable as we can. But just getting rid of it wholesale, I think. I'm not going to say too much about animal welfare, but I mean, I think one of the questions is ethically, should we kill animals at all? I think one of the things to be aware of is we did actually evolve as omnivores. If you look at our current diet and you look back at either hunter-gatherer societies now or the evidence from pre-agricultural societies, in fact, the proportion of animal protein in the diet was higher than it is now. So typically our ancestors actually struggled to get enough plant food, particularly the ones that were further away from the warmer climates. I think, having said that, I think we do have an absolute obligation to minimize the suffering caused by livestock farming, so both within the farming process and also at slaughter. And I like to think that some of our research has been supportive of that, because one of the things that we will talk about this soon is that any stress on an animal, any poor welfare, will lead to poor quality meat. So high quality meat, which I'm associated with, the high quality food requires high quality treatment of the plants that provide. Finally, I just want to briefly talk about health. There have been, particularly recently, there's been long-term issue around saturated fats and the connection with heart disease. And more recently, there's been reports, studies that come out linking red meat and processed meat with cancer. And they may increase the risk of cancer. I think large diets with excessive consumption of meat have got a correlation with higher levels of cancer, particularly bowel cancer. And that evidence is actually quite strong for reasonable amounts of processed meat. I think these studies are reasonable for Western populations like Europe, probably New Zealand, Australia, and America. And I think to some extent we could lower the portion of meat we eat. Most other populations in the world, they actually eat quite limited amounts of meat. Many of them would be better off, I think, nutritionally eating more. And we hear that people are eating less meat, but worldwide, I think the growth rate's been effectively 2% for something like the last 50 years. So production of meat is increasing, and certainly increased now last year. So I think there is good evidence for replacing the health benefits of replacing the current Western highly energy dense diet, meat as part of that, with a great proportion of plant foods. I am concerned about the suggestion that that's the same as replacing red meat with highly processed non-animal products. So there's a big difference between increasing the proportion of plant food in your diet and eating a plant-based meat alternative. I can understand why people would do it, but I think that's a very different nutritional equation. Okay, so what are we doing in this area? We'll go on to each of these anyway. So yeah. First up, our overall interest has been in quality of meat, and particular interest for us recently has been high pH in meat. The highest quality meat, New Zealand meat, goes away as chilled meat. Some of it has to travel to the UK, which is some of our highest. The UK and Europe are 10 of our highest premium markets. They spend a long time on that boat. And we've worked very hard to chill that meat and hold it at the temperature to give those multiple weeks and arrive in good quality. But that relies on the meat being of the correct pH. And so meat actually becomes more acid after the animal dies. And if it doesn't become acid enough, if it has high pH, which means it's not very acid, it will deteriorate quickly. And so we've been doing studies on trying to be able to detect that. I'm just going to go through the most recent experiments on that. But what we found when we tried to study this is, although a reasonable percentage of our meat ends up being high pH, it's really hard to tell when you go to the works which meat will be there. So we needed a reproducible model of producing high pH meat. And the way we did it was we took sheep, and in the 2 hours before they slaughtered, we just really effectively walked around the paddock once every 30 or 40 minutes. So we're not talking, sprinting. It's just moving, and that process of being moved rather than being able to just graze and do what sheep like doing was enough to give us a difference between them and the sheep that were kept separately in a paddock, just really quietly near where they're going to be slaughtered, to actually lead to a reasonable proportion of high pH meat. And this is what I show this picture, because meat actually does involve slaughtering animals. We try to avoid that in the meat industry. We try to show you that. So this is the experiments we're doing. We were actually looking at multiple muscles because you can expect different results in different muscles. So this was the whole sampling process so we could get the different muscles. And then we studied their meat quality characteristics. And as I said, what we did with this, we studied the quality of this meat, so its tenderness, its water holding capacity, its colour, all the things that we can determine what state the meat was, and naturally its pH. And we could show that exercise did lead to a consistently high pH. What we were interested in really with this, and this is the biochemistry part, is we wanted to know what was happening inside that meat. So what was the metabolism that was going on? So we used this machine here, which is called a rapid evaporative ionization mass spectrometry. And Alistair Ross, down the back, thanks, is in particular uses this really well, and he did this work. What this works, it originally was used by a surgeon, but basically using the mass spectrometer, we can detect proteins, but there's usually a lot of work to do before that. What happens with this is just using either a hot knife or we've gone on to using a laser. You can burn the protein you're interested in. So you can see the meat patty here. This one over here was done on milk powder or milk. What that does is causes volatile compounds. Those volatile compounds, which are the compounds in that meat, can then be detected in this mass spec. So you get a result within minutes. And using that, we were able to detect differences in the muscle, so we could actually identify which muscles, different types of muscles from each other. And for this particular experiment, we could separate out the animals that had exercise from the ones that had no exercise. So each of these spots, what we're actually detecting here are the different molecules inside the cell. So with this, we could start to unpack which molecules are changing as we do these things, so we can understand what's going on. Okay, so just briefly to cover another couple, because Tim's given me the thumbs up, I'm nearly finished. We also do a lot of work, this is an example of pulsed electric field, of using different machines to design new processing technologies for food, foods. Because particularly as we go to offcuts of meat, or if we go to new products, we're going to have technologies that will allow them to be more digestible and more palatable. So we're studying those. We've also done work to decrease waste. Because animal products have a higher footprint, we want to make sure we use 100% of the animal. And we do that in the industry anyway. Co-products in New Zealand are worth more than $1 billion. So that's the stuff that's not meat of our meat animals. We're also doing projects to try and so PhD projects have been involved in relatively recently, involved beef lungs turning into a protein-rich pasta, which probably won't sell in New Zealand, but would be a really useful thing in countries where there isn't enough protein. Proteins and peptides from oat bran, so side products from plant proteins, plant products. And most recently worked with Luca around, he's getting proteins and peptides out of the processing water from legumes. So each time trying to recycle those wastes and get as much back as you can. Also, Crystal's talked about this a bit, but this is her PhD, Crystal O'Connor's, looking at proteins from insects. So we are looking at a wide variety of other things. In most countries, the proteins from insects will be insects that live off waste. So the whole idea is in a continent like Europe, where you've got masses of waste, if you can use some insects, and like often in a grub form, you can actually turn those into protein that is edible. I think for New Zealand, that's going to not be a particularly viable approach because we're not going to have the amount of waste. And we've also always got to be export, so it's got to be high quality. So for us, it's going to be more important that we have products that actually are differentiated and can play off our clean green thing. So what Crystal's been working on is this kawa looper. So using the advantages of knowing that this is something that in Maori, that actually they have known for a long time that this plant is a very healthy plant. So getting that association and seeing where that comes through to the insect. I've also done some of this work in collaboration with target industries. And just to finish off, to give you an idea of the future, I suppose, project that I've just signed up to support is one at University of Canterbury, take with in vitro meat and combining it with plant protein. So in vitro meat is the idea of growing meat in vats. So using animal cells, so cells that are taken from an animal, growing them up using the technologies we use for bacteria and fungi. And it's got a low environmental footprint, you've got no farm, and of course has potentially the advantage that there's been no animal killed because you can extract these cells without killing the animal. And there's work going on around the world on this. We did a review of this back in 2019, and at that stage, there were quite a few, I mean, there was a lot of hype around it, but there were a lot of issues. And one of the first one was you actually require fetal calf serum, which is actually because to actually make these things grow, they've got specific things. Cell growth is actually quite difficult. And so that becomes a little bit dodgy if you talk about no killing animals. It's also something that's in very short supply. So as you scale up, it will be very difficult. There was also issues about whether it be regulated or not. And then greenhouse gas emissions, because these are all proprietary things, the guesses about greenhouse gas emissions are all over the place. People think there'll be no farms. There's a lot else going on when you start fermenting things. And there's also issues about trying to get it to be meat rather than mush. There's a lot of structural meat. And the big issue is scale up. Now, an indication since 2019, these first two things have largely been sorted. The people who are doing this have now got FDA approval, and they've tended to be able to replace those things that for a long time we haven't been able to replace with plant products. So the key thing with this is I think it's still got a long way to go, because the scale up in particular, getting the oxygen to those cells once you start trying to make more than a few burgers is going to be really, really difficult. But, you know, it's the interesting thing about technology is technology always gets better. So that would be really interesting to see where this goes. I could imagine it coming down to a reasonable price, and then it'll be interesting to see what people think of eating it. I think in all this, my take-home message is I still think that what New Zealand does, which is grow a specific natural product, there will be a market for a very long time for meat. But some of these other things may well be the solution to the question of providing a protein source for the people who can't afford that high quality. So I think we'll be targeting the higher end of the market, which is what we've always done. So I think, this is just to say that in a couple of weeks' time, I'm going off to the Meat Science and Technology Conference. There's one every year. This year it's in Italy, and it's just to remind everyone the meat in other countries doesn't mean what we think. So this is what they think of as meat. They, in the Italian, the types of meat they eat, they have down at the bottom 1% each of rabbit, lamb, and horse. So lamb is not on the menu. So I think that hopefully gets me. Oh, so sorry, of course, acknowledging, and this doesn't acknowledge everyone, but work's involved over the years, an awful lot of people over, Lincoln University staff typically here, and PhDs, some of whom turned into staff, and then a long experience collaborating with AgResearch and also University of Canterbury and University of Sydney. So thank you for that. And I think next sections. You answer the questions.
Oh, no, you answer the questions, Jim. And so there's an opportunity for some questions for Jim to be taken. I'll just ask that if you raise your hand so we can see you, and also just as you're asking the question, perhaps just give your name associated with that question as well.
Very interesting result about stressed animals and changes in meat. What's your chances of persuading the meatworks to change their behaviour?
The meatworks actually take that stuff really seriously, because high pH meat's a disaster for them. I mean, they have things that I know have changed in meat-works, because stress in animals is an interesting thing. So they try really hard, for instance, not to have dogs anywhere near them the night before. Having dogs barking nearby can be quite unsettling. They've also worked on trying to not have herds mingling. I mean, if you bring animals in that have known each other before, particularly cows or cattle, they will try to re-establish dominance. And so it's not a great thing the day before. But there's also odd things that people have learned, like cattle don't like traveling up steep angles. So the angle as you go up to slaughter is critical for cattle. So there's those sort of things they actually do take seriously because it impacts their bottom line. So if you can show that research, you can make a difference.
Thanks,
I
Jim.
can from Link University.
Yeah, just on the exercise thing, that's really interesting. So, have you thought about a mechanism there? Is it a change in cortisol or?
No, the mechanism largely is around glycogen and glucose. So, I mean, the actual exercise model was important for us to get the high pH. But the work around why you get high pH is largely known. So it's if the animal either hasn't, so when the animal dies, the muscle doesn't know the animal's dying. All it knows is all communication's gone to the rest of the world, including its oxygen supply. There's no blood flow. So it continues to operate. The cells will go as long as they possibly can. And they've got their own supply of glucose in the glycogen, so they'll start breaking it down. So two things can happen. Either in what you want to happen is you get down to about from pH 7, which our muscle normally is, down to about pH 5.5. And at that stage, the enzymes stop working effectively. So everything stops, and that is what we would normally think of as good meat. The other thing that can happen is if you've used up your glycogen already, then there's not enough there. You run out of fuel, and then you get stuck halfway down. And that's what's happening there because they're mobilizing that glycogen just the same way as we were quite surprised to get as much effect though, because you wouldn't have thought they would mobilize all of it that quickly. You can also get the same effect if you don't feed them well before they go to the works, because then they haven't built up the glycogen.
Travis Ryan, something.
Jim, from a commercial perspective for New Zealand, if we were to go more down the alternative or plant-based protein pathway, do you see us being competitive in that area in the future? Would we be an exporter of the products or an exporter of the technology that produces the products?
Plant-based proteins, I think there was a really good survey done by Plant Food. They had to do a report on it. The issue for us is we're very unlikely to be able to compete against the Canadian or the South American plant protein crops. We're just never going to get the acreage. So, I mean, I can't see us being able to compete directly in producing those plant proteins. We could produce products as technology, but not. That would be my view. Just the scale of those. And then that means, of course, they have the big factories.
Aaron Lucock.
Very interesting presentation, Jim. I'm interested in knowing more about that Calpine triggered tenderising process. Has there been further research done on consumer perception about the cows, the not so great meat being tenderised into acceptable meat versus the top quality cuts? Is there any research done on consumer?
You mean
perception?
in terms of how?
Yeah,
If
what
they're
they have
told,
done, yeah, blind testing to whether they can tell the difference.
well, no, that it was done in that work. Yes. So I showed you the actual machine tenderness, but the consumer panels really couldn't pick the difference. They saw it as tender, and they liked the texture equally between them. Because we were, especially once we saw those diagrams, we were a bit worried that we had mushed it too much. It's quite hard to tell which, because that structure that we saw in that diagram, that's why it's much easier to make a mince, textured vegetable protein than it is to make a steak. There's an awful lot of structure in a muscle. And it's in two ways. One is the actual protein structure that's all there, which you were seeing at a microscopic level, but they're large proteins you look at. The other is the fat is actually contained in globules. So when you chew into them, that way it comes out. It's not the same as just trying to mix oil with protein. You'll get a different, your mouth feels quite different.
Opportunity for one last question.
Hi, Chris Morton. Just curious about whether the breed of animal or the type of livestock has an impact on the quality, and also if any of this research has gone back to the farm and are farmers changing anything about the way they do because of the research you've been doing?
The breeds do make a difference. Most of the so meat breeds often do show up better in those taste tests. It's more a taste testing thing than what we do. You do see it in the extremes. There are, I mean, in beef, the difference between Bos taurus, the beef that we normally have, Bos indicus typically always comes out tougher. So you do get differences like that. The question about most of our research has tended to actually lead back to changes at the later ends of the thing. So our work hasn't tended to affect too much what farmers do. It has sometimes affected what Silverfin Farms has done. And also it's certainly that picture I showed with Roy Bickerstaff was part of a project where he was part of that New World Guarantee scheme on the meat. And he found when he started doing those surveys of New Zealand meat, the issue was that obviously the meat that goes onto our market comes out of the same, well, typically now comes out of the same plants as goes overseas. But when it goes overseas, it's chilled and it goes through a whole process. When it was coming here, it goes to the supermarket and then they have to age it. No, they were cutting corners. So back in the late 90s, New Zealand was actually pretty damn tough. And it was because they weren't aging it. The supermarkets got really good at doing specials. And when they got specials, they'd be going to the works and going to shelf the next day. And it takes 5 or 6 days to age the meat. And that's all time that's sitting in the chiller. And they weren't doing it. But once we started surveying it, they started doing it. And it improved dramatically over that 5 years. So I'm not sure that we can claim much back to the farmer. We can claim for the consumers it did make quite a difference there. But interesting, I mean, that is largely a matter of just testing the quality and then.
Well, thank you. Thank you, Jim, for that presentation, taking us on the journey from barley rust through to through Calpines, Cataracts, and on to meat quality, and a journey that has taken, I'm sure you would have seen through many collaborative partners and many students on that journey, and their contribution is recognised here as well. Just a small sign of appreciation here. So this is your blanket in here, which is the Lincoln University one, which I think you may need tonight. So thank you very much. Towards to the end, this part of the proceeding. So there are some drinks and light refreshments just round to the corner, out here to the right. So thank you very much.